Nucleic acid structure and DNA case study by Zohar San and Milagros Santos. For this case study, our job was to imagine that we were members of research teams that focus on genetic information systems and samples collected from Mars by a probe that has just returned to Earth. Initial analysis showed us that in the sample there was eukaryotic unicellular life. It also indicated that in the presence of double-stranded nucleic acids that were similar but not identical to DNA found in eukaryotic organisms on Earth, the alien nucleic acid had two strands of nucleotides bonded together with the sugar phosphates on the outside structure. The alien DNA had 6 nitrogen bases, which we can look at here. 6 nitrogen bases for alien DNA are isoadenosine, uracil, isocytosine, isothiamine, beta-guanine, and adenine. They're located on the interior of the double-stranded nucleic acid. The data also suggested that the complementary base pairing between the nitrogen bases of the two strands is that isoadenosine is complementary with uracil, isothiamine is complementary with adenosine, beta-guanine is complementary with isocytosine, and this is different than Earth DNA because in Earth DNA we have a 5-carbon sugar and a nitrogen-containing base that's attached to a sugar and a phosphate group. Also, our DNA has different pairing rules. Adenosine pairs with thymine, cytosine pairs with guanine. Again, both the Earth DNA and the alien DNA have similar double-stranded nucleic acids. Alien DNA has 6 nitrogen bases. Earth DNA has 4. So after this, the alien DNA was brought back to the lab and a double-stranded piece of nucleic acid was isolated. And it was found that each individual strand of the nucleic acid was 160 nucleotides long. This made it a total of 320 nucleotides. Out of the 320 nucleotides, 95 of them contained uracil. So knowing this information, we were able to answer the first 4 questions of part 1 of the study case. So here we have question 1 that asked us how many nucleotides containing the base isoadenine would be found in the isolated piece of the double-strand nucleic acid. We said 95 would contain it because in the information given, it told us that out of the 320 total nucleotides, 95 of them were uracil, and we know that uracil pairs up with isoadenosine. So therefore, 95 would also be isoadenosine as well. The second question was similar, and it asked how many nucleotides in the piece of DNA, in the piece of alien DNA would contain the base adenine. So for this one, we can't really determine how many. In the first question, there was 95 uracil, and we know that uracil pairs up with isoadenosine, but since we only have 6 pairs and a total of 320 nucleotides, we can't really determine how many adenines there are. We only know there's 95 uracil and 95 isoadenine. The third question asks us, if it were determined that the sugar phosphates making the nucleotides in alien nucleic acids were similar in nature to those found on Earth, how many phosphate groups would be found in the piece of DNA? And we said that there would be 324 phosphate groups because each nucleotide in Earth DNA has 1 phosphate group per nucleotide, and on the 5' end of the DNA, each DNA strand has 3 phosphate groups instead of 1. Resulting in 2 extra phosphate groups per strand, so it's 4 altogether, plus the 320, making it 324. And similarly, this would mean we have a total of 324 phosphate groups in the alien DNA also. The fourth question asks us, which set of experiments discussed in this chapter would be most useful to determine the replication method utilized by alien life form, and explain how this experiment works. Include the experimental design. We said that the Michelson and Stahl experiment would be most useful. So the Michelson and Stahl experiment would be most useful on how DNA replicates. These 2 researchers wanted to figure out how DNA replicated, So they created 3 models on DNA replication. These 3 models are conservative, semi-conservative, and dispersive. The first model here is conservative, and it means that the DNA produces one double helix with both parental strands and the other one with 2 new daughter strands. Second one is semi-conservative. In this method, a DNA molecule with one parental strand and one newly made daughter strand are produced. And this last one here is dispersive. This happens when DNA replication produces DNA strands in which segments of new DNA are interspersed in the parental DNA. Here is the experiment. So first, the bacteria cells were grown for several generations in a medium that contained a heavy isotope of nitrogen. And then the cells were transferred to a new medium containing a lighter isotope, N14. At various times after the transfer, samples of bacteria were collected. The DNA was then extracted and dissolved in cesium chloride and then spun rapidly in a centrifuge. When cesium chloride is centrifuged, a concentration gradient is made in the tube. DNA moves in the gradient until they reach a place where their density equals to that of the cesium. DNA that has N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that of N14, so it sank to a lower position in the cesium gradient. And then they looked at the DNA under UV light. Here they found the results. After one generation in the N14 medium, bacteria yielded a single band of DNA, indicating that only one strand of each duplex contained N15. After 2 generations, the N14 medium had 2 bands, one intermediate density and one of high intermediate density and one of low intermediate density. In the end, they concluded that replication of DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form complementary strand on each of these templates. They found that the Earth DNA replicates with the semi-conservative method, so we can do the same experiment with the alien cells rather than the bacteria cells and conclude which method of DNA replication is used in the alien cells. So if you look at the picture in the case study, it seems that the first generation of replication had a mix of old strands and new strands. So when we replicate it, one band should be the result. Through this, we can automatically rule out conservative replication, and then after the second round of replication, it should still weigh the same. So aliens replicate using the dispersive method of replication. This brings us to the second part of our case study. First question asks us, examine the structures of the genetic material of the alien sample, explain the similarities of those with Earth DNA. So from the information before, we know that both have a double-stranded nucleic acid and share the base pairs adenosine. Their base pairs are also made of nitrogen and both have sugar and phosphate groups. Second question asks us about the differences between the two. We know that the alien DNA has 6 nucleotides instead of 4 like what the Earth DNA has. Alien DNA also replicates through the dispersive method rather than the semi-conservative method like Earth DNA. And Earth DNA runs anti-parallel, alien DNA does not. The third question asks us, the alien DNA it asks us, compare and contrast the process of replication between the alien sample and the eukaryotic life found on Earth. So alien DNA replicates through dispersive replication because, like mentioned before, the first generation had a mix of old strands and new strands. So when we replicate it, one band should be the results, ruling out conservative replication. After the second round, it should still weigh the same. So Earth DNA replicates through the semi-conservative method. Alien DNA replicates through the dispersive method. Semi-conservative method involves one parental strand and one newly made strand being produced. Part 3 asks us, for this part, we were told that the piece of human double-stranded DNA was isolated And in the piece of isolated DNA, there was 410 nucleotides. 88 of them were adenine. So the first question asks us, how many phosphate... First question. Sorry, my bad. Okay, the first question asks us how many nucleotides in the piece of DNA would contain the base guanine. And we said 117 because there's 88 adenine, and adenine pairs up with thymine, so 88 and 88 is 176. Then we subtracted 176 from 410 and got 234. And half of those are guanine, meaning there's 117 guanine nucleotides. Now to this question, which asks us how many phosphate groups would be found. We said there's 414 phosphate groups because each strand has 205 nucleotides and each one contains one phosphate group. That excludes the nucleotides at the 5' end, which carry 3 phosphate groups each, so we would have 205 nucleotides on each strand, making it 410 altogether, and each strand has 2 more phosphate groups at the end, having 2 strands, 4 plus the 410, making it 414. The third question asks us, we know how many molecules of uracil would be found in the piece of DNA. For this one we said 0 because human DNA does not contain uracil. That is only in human RNA. Question 4 asks, in the DNA bonded molecule, which are not bonded together? So out of the 5 choices here, we said the phosphate groups and the cytosine are not bonded together because in human DNA, phosphate groups are not attached to the nucleotide, so that cannot be it. Adenine and thymine, we know for the DNA base pairing rules on Earth, this is how they pair up. Deoxyribose, the sugar and the phosphate groups do pair together. Cytosine and guanine, again, base pairing rules, they do bond together. Deoxyribose and adenine, the sugar can bind to the nucleotides. It's just that the phosphate groups cannot bind to the nucleotides. And last question, number 5. Given the original piece of DNA shown to the right, which sets of DNA below reflects the process of replication as it occurs in eukaryotic cells? So here's the picture to the right, and it's asking us which one shows the process of replication that happens in the Earth DNA. So here's the picture that was shown to the right. This one, it seems that there was 2 new strands that were made. This is the conservative method, and Earth DNA does not replicate via the conservative method, so we can automatically rule out this A. B, this has a mix of old and new. This is the dispersive method, so we can rule this one out too because human DNA does not replicate through this method. This one we said was wrong because it does not run anti-parallel. It's 3' end to 5' end, 3' end to 5' end, rather than 3 to 5 and then 5 to 3. So we know that this is not an option. Finally, we said that D was the correct option because human DNA replicates through the semi-conservative method. And it is shown here that this is the semi-conservative method, so we already know that this would be the one that replicates in human DNA. And that's it. Thank you for watching.